This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 68, recorded on March 5th, 2014. Hey, everybody. I'm Vincent Racaniello, and with me is Dickson Despommier.
Hello, Vincent.
Why are you smiling?
Oh, because I'm having a good time.
I could make you not smile.
I know. We don't want to go there, do we?
We have a guest today.
We do.
Right here in the TWiP studio.
This is very true. There are now three of us.
Outside of which it's a cloudy day.
That's true. Also true.
His name is Sagi Shapira. He's a professor in the Department of Systems Biology.
Systems
Welcome,
biology.
Welcome, Sagi.
Nice to be here.
Which systems are we talking about, Sagi?
Systems of biology.
The systems. All of the systems.
That's right.
Okay, fine.
Where are you from?
Israel.
And then you moved.
Originally from Israel, grew up in Europe.
What
Africa.
part of Israel?
Originally from Haifa.
Haifa. Oh, that's interesting. Haifa. That's a different
Then
city.
spent time in Germany and Scotland, then Kenya.
Are you multilingual?
Some say. Partially.
Yeah.
Give us an example. No, don't do that.
You got a PhD at UPenn, right?
That's right.
You actually worked on a parasite.
I did. I worked on toxoplasma.
Do you know Phil Scott?
I actually spent a lot of time with Phil Scott.
He's a good friend.
I was in the lab of Chris Hunter.
Chris Hunter.
Studied,
Did you know Michael Phillips?
I do.
Okay.
You studied Toxoplasma gondii, right?
Toxoplasma and how toxoplasma interacts with the cell's signaling pathways.
It's a very interesting parasite. We've had John Boothroyd on as a guest here, by the way.
Actually, Chris Hunter, his supervisor, was here giving a seminar.
Really?
And
I
recorded...
Very entertaining guy.
I recorded a 15-minute conversation with Chris, which I never released. It's too short.
Oh.
Would have to be appended to a TWiP, you know?
True.
But, yeah, I should do that, shouldn't I?
I think it would be fascinating.
Then you left Parasites. You did a postdoc in...
I left parasitology, and I did a postdoc, a very short one, with Alan Izigowitz at Harvard.
Which field was that?
In innate immunity,
okay.
working in Drosophila.
All right.
In Drosophila phagocytosis. And then after 6 months, moved to another lab with Nir Hakohen at the Broad Institute and studied viruses.
Really?
Another favorite.
Back to parasites.
That's right.
Dixon, listen to this.
I'm listening.
Are you a virologist?
I use viruses to study innate immunity.
Okay.
Is that a yes or no?
I wouldn't be brave enough to commit to defining myself.
To me,
as a
he always
virologist.
says I'm not a virologist. Now you are here. You have your own lab here at Columbia.
I do.
In this brand new department, Systems Biology.
That's right. Very exciting.
And you still work on viruses, but with,
we do still work on viruses, and there are two main interests in my lab. One is how the innate and adaptive immune system influence and provide selection pressure to influence viral evolution. And the other part of the lab is interested in how cells make decisions. Cells of the innate immune system make decisions. How they turn on, decide what genes to turn on, or other genes to turn off.
It's a good thing you're not studying Dickson because he can't make any decisions.
Stop it. Of course I can make a decision. I decided to have this TWiP today, for instance.
Okay.
And you chose the paper.
And I did choose this paper because I thought it was...
Dixon, you need to stay near the mic. I can hear you fading.
It was a follow-up on a...
You're going back to the boom. I'm sorry. You're being demoted.
No, you didn't let me finish my sentence.
I love
we chose this paper. Sagi has to know why we chose this paper. Because we've been talking about sex and parasites for a while
now. I haven't been talking
now.
about sex. No
sure
way.
We did. We've covered it in several organisms.
Which
already.
parasites reproduce sexually? Does toxo reproduce sexually?
No.
No? What about...
Really?
Sexually?
Really? What's the definitive host for toxoplasma?
And the kitty cat.
Right. And what happens in the gut tract of the kitty cat?
It's a good thing Chris
I might
Hunter
have
is not
caught
listening to this.
That's right. You don't do parasites anymore. No problem.
So for toxo, they do have sexual reproduction.
Of course they do.
That's true.
And
It's
they
a,
produce as their stage in the gut tract of the cat only.
That's right.
The stage is called the...
Oocysts.
It's called the oocyst.
Oocysts.
That's correct.
there,
And so
there are
the
male and
oocyst-
female oocysts. But not really
there
male
are.
and female.
No, I think there are male and female parasites. There has to be
For toxo.
something like that. The same is true for Cryptosporidium. Same is true for all of the apicomplexa.
Okay.
So
That's
that's
right.
toxo is
That's
an
right.
apicomplexa,
You're
right?
taking
So
it back about 15 years.
exactly right. So it's okay. This is old literature, but it's still valid. So today we're going to talk about a parasite that reproduces sexually inside the salivary glands of its vector. We've already discussed one, the most widely distributed, I think, and most feared protozoan parasite, at least, malaria, which has sexual stages that take place in the gut tract of the mosquito. Of the female mosquito. That's right.
Right.
So,
Trypanosomes were not known to reproduce sexually.
well, they were suspected, though, weren't they?
It was suspected, and this is a paper that provides some evidence of
Exactly.
that, right?
Exactly. In fact, they've got very good evidence that it does occur because they've got visual proof.
So the name of the paper is Meiosis and Haploid Gametes in the Pathogen Trypanosoma brucei.
Right.
It's published in Current Biology, and the authors are Peacock, Bailey, Carrington, and Gibson from the UK.
Exactly.
And one of the institutions is the University of Cambridge
Right.
and the Department of Biochemistry. Famous,
Very famous.
august.
Do you know who the most famous person that ever worked at Cambridge was?
Was that Sanger?
No, it was
Max-
Isaac Newton.
These guys are more famous than
Sir.
Isaac Newton. Sir Isaac Newton.
Sir.
Didn't
Isaac
Max
Newton.
Perutz work there?
Yeah, he did.
Didn't Sanger?
He did.
I love where it's located.
Stephen Hawking's is
He's still there?
Yep, sure.
Oh, what's his title, by the way?
Hawking.
I think he's the Isaac Newton professor
Yes.
of astrophysics.
Yeah. Isaac Newton. Wouldn't you love to be the Isaac Newton professor?
Well, you remember what his father did for a living, right?
No,
'Cause
he's the
we've covered
Lucasian
this.
professor.
Is he really? Okay, fine.
Who was Lucas?
Do you know who Stephen Hawking's father was?
I do not.
He was a parasitologist.
No
He
kidding.
worked on worms,
What kind of worms?
filariasis.
You know, Britain has a very long history of,
Frank
of,
Hawking.
of,
That's
of
exactly
studying
right.
parasites.
Exactly right. In fact, the former dean of the vet school, Lawson Soulsby, was the former chairman of the pathobiology department at the University of Pennsylvania, where you got your degree. And that's how come I got to know all those people, because I used to go down there a lot to give lectures.
The
and
Lucasian professor.
Lucasian.
Okay, who was Lucas?
No idea.
You don't know? Okay.
Nope.
All right, this paper. How did we get on Steve?
We got
Hawking?
from Cambridge.
Well,
We were
I didn't
just
finish
covering
saying
some-
what I wanted to. You just interrupted
Sagi
me.
is now getting the impression that this is not just about parasites.
It is. The University of Cambridge is on Tennis Court Road, which I always liked.
is.
I remember seeing that address when I was in graduate school.
Okay.
Wouldn't you love to work on Tennis Court Road?
I would.
Do you play tennis?
I used to. I was on my college team for a while.
How about you, Sagi? You
I
play
got kicked
tennis?
off.
Did.
You used to play tennis?
I played varsity tennis in high school.
Did you really?
That's right. And actually, my high
Yes?
school has the longest standing record for winning New York State.
I'll be darned.
Yeah.
Have you
And
produced
they-
any
and they have even
professional
won
tennis
nationals.
players?
So you went to
Yes,
high
they
school
have.
in New York State?
Yeah, in upstate New York, in Syracuse.
Would we know any of those people?
Syracuse. Wow.
I'm not sure if I know any of those people.
All right. So you picked this paper,
I did.
Dickson.
I did.
And we're going to go through it, and Sagi will give us his sage.
Exactly. He did a quick
Advice.
read, but I think because of your acumen in molecular aspects, you were going to give us some insights which we wouldn't ordinarily have
Don't
acquired.
expect too much.
Sagi is a critical young man.
Indeed.
He has a sharp eye.
It's the only way to get ahead. You have to be skeptical.
Now, Dickson.
Don't expect too much, but I do want to correct one thing you said,
Uh-oh.
which is initially you said trypanosomes replicate in mosquitoes, but
No, I never said that.
I thought that's what
No.
I heard you
I
say.
said only
But
the apicomplexa, only malaria was the
yes, okay.
Where did trypanosomes replicate?
one that... Right. In the tsetse flies.
Tsetse fly.
Yes. And
Reduvids? No.
Yeah.
Right.
They're
Yeah,
South,
no,
they're
that's
South
kissing bug.
American.
That is
The kissing
exactly
bug.
right. That is exactly
The South
correct.
American ones, right?
You're right. So those are the two vectors for American.
Reduvid bugs in South America, Africa,
Correct.
tsetse flies.
That's right.
What a great name.
Right.
You take the E out, then you would have no vowels. Flies.
That's right.
All right, so it says here, Dickson,
Yes.
meiosis-specific genes have been identified in Giardia and Trypanosoma brucei.
Right.
So meiosis, what would meiosis have to do with reproduction?
Or anything for that matter. In fact, meiosis is the only way to get recombination
tell our
between two
listeners.
different.
So if you want to make gametes, you have to have
Organisms.
meiosis, right?
That's right. It allows for the best way to exchange genetic material.
So what does meiosis do?
It reduces the number of chromosomes to 1/2 of the amount that was there
Okay.
before.
That way, when the two gametes come together,
They
you have the
can
normal
restore
number.
the
Because if you didn't
diploid
do
state.
meiosis, you would have too many chromosomes.
This is true. And are there organisms that have too many chromosomes? Normally?
I know a few of them.
I can tell you lots of them.
Go ahead.
It's called polyploidy,
Yes.
right? And plants in general have huge numbers.
And they can
of-
deal with this?
Apparently,
Because HeLa cells are aneuploid. Do you know what that means?
yes.
They have more
They have
than
a-
46, but every cell has a different
You're
number.
exactly right. And they have trouble, don't they?
HeLa cells are fine with me. They grow polio really well. I don't have a
I mean,
problem.
none of the lost chromosomes
I
would
think they
affect the
became
outcome.
aneuploid in culture over the years.
Got it.
And but polyploidy means you have more than the N.
That's right.
So for humans, what is N? 23?
yes.
And we
Pairs.
have, and then in
Pairs.
our
Mostly
somatic
pairs.
cells, we have 2N, which is 46.
That's right.
So meiotically derived gametes would have 23 or
Exactly.
N.
You know
right?
why I know that? Because there's a site that you can go to now and determine your genome, and it's called 23andMe, and the 23 is based on the number of chromosomes you have.
Are we okay, Sagi? You got to make sure we're saying the right things.
I'm checking on it.
So far, so
Everything-
good.
Everything we have said so far is correct.
It's all valid.
All right.
Okay.
So the fact that they discovered these genes, by the way,
Some
they-
amoebae, by the way, just to interrupt, have huge numbers of chromosomes.
this is a question for Sagi. They say
Now,
they
of course, there are human diseases that are associated with having multiple copies
Right.
of particular
Or-
chromosomes.
Exactly
Right?
right. The duplications,
And
etc. Yes,
most
indeed.
most famous
the
one
most
is
famous is Down syndrome.
Right,
Trisomy
trisomy
23.
23, is it?
I think so.
21.
21,
exactly,
trisomy 21.
Exactly.
All right, Sagi, they say in this paper, meiosis-specific genes have been identified by phylogenomic analysis. What does that mean? Phylogenome.
It's a systems question.
Do you like that
I'm
word?
not sure if we probably don't want to get into discussion of how we define systems biology, but
Is that a valid term,
That's for a
phylogenomics?
different show. But what they're trying to do is the concept is that by looking at genomes of different members of a particular species
Right.
or across different
Right.
species,
Right.
we can learn something about how related they are.
Okay.
And by aligning these sequences, we can find regions of the genome that are either similar or different,
Right.
and we can then infer either function or shared ancestry.
So if you have
So-
genes that are known to be involved in meiosis in one organism, then you can look at related organisms and say, these might be
Right.
involved in that function, right?
Exactly.
And that's what they did here.
Right.
So they have 3 genes called MND1, DMC1, and HOP1. And these are all in Trypanosoma species, and they're supposedly meiosis-specific proteins.
So
Okay.
they say.
And in this paper, they're going to look at that. So that's where they first got these meiosis-specific proteins. So
Right.
the fact that they're there suggests that these organisms undergo meiosis, which suggests
That they have sex.
exactly. Now you got my cue right on. It's
I
great,
did.
Dickson.
I did. I'm a perfect foil for your cues.
So I didn't know you could do this with trypanosomes. You could put genes in.
Oh, yeah, you can transfect.
Can you?
One of the beautiful things in some of these models, many of these parasites that people have developed genetics.
Right.
Done it for toxoplasma, trypanosomes,
Yep.
malaria.
Yep.
That's what makes these things very tractable experimentally.
So can you modify the gene in situ? Can you replace a gene with a tagged version, for example?
I think that I haven't followed the literature for trypanosomes, but depending on the parasite, it's more or less possible.
So in this case, what they did is to put they used yellow fluorescent protein tags on each of these meiosis-specific genes and put them in the trypanosomes. And then they say that you can see these genes during in the salivary glands.
Right.
Now, what's the significance of that, Dickson? Why salivary glands?
Because the salivary glands, that's the site through which the vector releases the parasites. Right? And by the way, we should clearly distinguish those two groups of trypanosomes that we just mentioned before, the reduvid bug-transmitted parasites and the tsetse fly-transmitted parasites. When Pangea was connected, and as the Jurassic period developed 175 million years ago, South America and Africa began to separate.
Right.
And it's presumed that at that moment, these two groups of trypanosomes, which used to be one big group, presumably started to divide off into two groups. Now we have two completely separate groups.
What are they?
South American
brucei?
trypanosomes or American trypanosomes can be transmitted by tsetse flies, and the African trypanosomes
is that right?
cannot be transmitted by reduvid bugs. It is right.
No, so people have actually shown that they are they're genetically related, and that their
kinetoplastidae,
their last common ancestor was 175 million years ago.
They haven't gone through that machination yet, because how would you do that?
Well,
there's no remnant 175 million years ago. But the assumption is that as these two continents pulled apart, then that's the Darwinian concept of how you get speciation. You separate the geographic regions for the same species, and now all of a sudden you get two different species, right? The tsetse flies obviously didn't transmit across. Maybe it was a different vector even then than it is now. But eventually you've got two completely different sets of trypanosomes involving two completely different sets of transmission mechanisms. One is the bite of the fly. But the other one is a defecation event on the surface of the skin during the feeding episode that the reduvid bug goes through. So those are two completely different ways of looking at this, right? So if you say that mating in trypanosomes in Africa occurs in the salivary glands, well, you wouldn't look in the salivary glands of a reduvid bug for the same events for the American trypanosomes. Where would you look?
The gut.
Exactly.
Because that's how they release it.
Exactly.
So in this case, they're using T brucei.
Yep.
So the host is a tsetse fly.
Yep. Well, they're African. They said they were looking at the African.
Do they actually use tsetse flies for these experiments?
They could.
Let's see at the figure legend here. Figure 1.
There are colonies.
Part of a tsetse salivary gland. Yeah.
Yeah. No, there are colonies of tsetse
Wow.
flies. Not many, but there are. There used to be one at the University of Massachusetts.
So they see in the tsetse gland of an infected tsetse,
In the salivary gland of the tsetse
they
fly,
see
yes.
yellow,
Right.
which means these meiosis-specific genes are being expressed there. So they say this is the first time there's been evidence of meiotic cells.
At least we found the location where this is occurring.
In the salivary gland.
That's
now,
correct.
the interesting thing here, this is a little aside.
Go on.
They say sex provides the opportunity for new strains to arise by recombination. And they make the point that in T. rhodesiense,
Yes.
it can infect humans by acquiring just one gene. So I didn't know it couldn't infect humans. T. rhodesiense. How do you say
Let's
it?
say this again.
T. rhodesiense. Rhodesiense.
Named after Rhodesia.
They don't infect people.
That's not right.
It says here they infect people when they acquire a single gene for human serum resistance.
Yeah. Oh, okay.
And that could acquire by sex, right?
Okay, that could be acquired by sex. That's right.
All right.
But it's interesting that there is a difference between West African trypanosomiasis and East African trypanosomiasis. And apparently they haven't crossed over to exchange genetic material because what do we know about West African trypanosomes? That there are very few reservoir hosts.
Mm-hmm.
Very few. And what do we know about the East African trypanosomes? They infect all the mammals, every one of them. So there's a huge difference here, right? And so Bruce, when he first started this experiment of trying to find out what the difference was, could not tell them apart morphologically. And so we had to wait for modern genetics to catch up to see what the differences were.
But now it's very interesting because the definition of a species to some degree is related to the ability to reproduce.
Yeah, sure.
And to undergo, at least in this case, sexual reproduction.
Absolutely.
So if Rhodesiense can acquire genes from
Rhodes,
from another species, right?
Yeah, then it suggests that they do undergo sexual reproduction, and perhaps the definition of species is a bit
it's
fuzzy.
such a gray zone when you get down into this level of what a species is. It's really no one has ever truly defined what a species is to begin with. So
in virology, we have species, but
well,
it's meaningless because they don't mate, right?
That's right. It's true. But even if they do mate, is what Sagi is saying, you still got a gray zone. Right?
Well, maybe you can exchange genes without mating, right?
Maybe.
Yeah, potentially.
So
who would do that?
I don't know.
The viruses.
Yeah. You could, I don't know at a viral level what mating would be, but if two viruses infect a single cell, they could exchange genes, but you wouldn't get hybrid offspring.
Well, perfect example, influenza.
Weigh in on this.
That has a segmented genome.
Yeah.
So if two viruses infect the same cell, then the progeny can have segments from either of the parental strains.
That's right.
And
it's like sex.
It's like viral sex.
Viral sex.
Yeah.
Sex went viral.
Sex going viral.
They look at a time course and look at when these genes are expressed in the salivary glands, which they can do by the yellow.
They can.
Racaniello.
Racaniello, that's right.
And they say the expression peaks at day 20. They see it between 14 and 38 days. And they say this is the right time.
Right.
But then they would like to find these cells that are actually gametes, right?
Yes.
And they have a hard time using flow cytometry, so they have to figure something else out, okay?
Right.
They actually say that there's too much noise in their experiments. There was too little cells, so they tried something else.
Right.
So what they do is they label separate. They have a red expressing trypanosome and a green expressing trypanosome, and so they can purify these putative gametes by red or green, and then they mix them together.
Ooh.
In vitro, not in a fly.
No.
Because the problem in the fly, they couldn't track, right?
Right. You'd have to get them to feed on equal amounts of both red and green to begin with to see that happen in the gut tract.
Did we say that?
Salivary gland.
Did we say that this shows that it's the salivary gland where the gametes form? Yes.
Well, that's where the genes are expressed.
It's not in the gut.
No, not
they're
in the gut.
They're in the salivary. Yes, that's where the genes are expressed. That's a good point because we haven't proven that anything's a gamete yet, right?
Exactly.
Right.
Exactly.
So they have two parental clones, one red and one green. They take these salivary gland cells and they mix them.
Right. The promastigote-like cells, as they call them.
They call them promastigote-like cells. They do this 20 times.
Right.
Right? They see small clusters of trypanosomes, so they tend to stick to each other.
Right.
They interact. They observe to interact by intertwining.
Which is not a normal way for trypanosomes to interact in vitro.
No,
if you grow
it's
them
not.
In vitro, they don't do that.
So these are putative gametes,
These
right?
are special.
You know, the funny thing is they look a little bit like sperm.
Well,
they have a picture. They have the thicker body, so they have a head that's very sperm-like. Right. Although the flagella is much thicker than a sperm would be.
Right. But you wouldn't get mating between two sperms, would you?
No, of course. That's the weird thing here is that these guys are mating.
That's
well, we don't know if they're mating.
We don't know, but they are.
But they're touching each other. Their flagella intertwined, Dickson. I don't know. That's a little NSFW, right? You know what NSFW means?
Not significantly.
No, not safe for work.
Oh.
You're really out of it.
I totally am. I'm an old school
You're like from
person.
another era.
I'm an old school person.
NSFW is what you label a website when you can't look at it at work.
Oh, I see.
Do you know that, Sagi?
Of course.
He knows. He's a
I
young
don't
guy.
look at those
invented it.
websites anyway.
Look at me. I'm an old guy, and I
looking
get
at
this
you.
stuff.
You're
What's
not
your
that old.
problem?
I have a bigger problem.
What?
I'm computer challenged.
Man, it's not like I've not tried. Anyway, these red and green cells, they touch each other. They intertwine their flagella.
They do.
And
Sounds
what
like
happens?
a scene out of a Voltaire.
Then
play.
within 30 minutes, they see yellow ones.
Yeah,
Single cells that are yellow.
right.
You can see that in the pictures. What does that mean to you, Dickson?
Well, if you have a yellow parasite and a green parasite and you mix them
No,
together.
green and red.
I'm sorry, a green and red parasite, and you mix them together and you get yellow, it probably means that they've fused.
That's it. So they actually say here, these signify fusion.
Right.
Now, how do you know it's
So
just
you agree
not...
with this?
How do you know it's just not de novo synthesis?
Exactly. Or, they just fused anyway, but it didn't mean that they exchanged or mingled their chromosomes necessarily.
They say that it's,
Right, but the suggestion they make is that because it happens so rapidly,
Yeah, right.
it's not.
it's not just de novo synthesis.
They saw each other from across a crowded room, and the next thing you know, they were together.
So they say these have a distinctive morphology. They have one kinetoplast and one nucleus, or two kinetoplasts and one nucleus.
So what's a kinetoplast?
That's it. Well, I understand it's a DNA structure associated with the mitochondria.
Yes.
Is that correct?
Yes,
So that's mitochondrial DNA, basically, right?
basically.
So if you
Well,
have two, no,
Well, there's other things there too, though.
it's not mitochondrial
There are
DNA,
many circles and maxi circles in each kinetoplast,
right?
and each of those sets controls different aspects of the respiratory tree for the organism.
Is that right?
But are trypanosomes aerobic or anaerobic?
But now, before you get to that, isn't there some idea of where kinetoplasts came from?
That I would not know.
I thought, and I may be completely wrong, that similar to the way apicoplasts were acquired, and same as
Right.
mitochondria, was through some secondary endosymbiosis.
Okay.
That
Okay.
it's a very ancient.
So we're hearkening back to the Lynn Margulis approach to the arising of symbiology, which is where does the nucleus come from? Where does the chromosomes? Where do these things come from? Right. So I don't know the history of the kinetoplast in terms of ancient history. Sorry.
So anyway, they say these look like promastigotes. Now, what is a promastigote, Dickson?
Well, it's one of the stages of development of the trypanosomes towards infectivity,
And this happens in the salivary gland.
apparently.
So
I'm not
would-
as familiar with the trypanosomes in tsetse flies as I am with the reduvid bugs.
Should we get the
because-
book?
Well, it probably wouldn't be in there because it's a general text for clinicians, and it's really not intended.
So the promastigote would be a precursor to a gamete.
Um,
Would that be the idea?
no, I think the promastigotes are preludes to the epimastigotes, which are the stage before the metacyclic trypanosomes.
Let's go to the book.
We don't really cover that well in this general text, unfortunately.
I don't know. You have some nice pictures in here.
We have some nice pictures, and we don't have any pictures from the salivary glands, though.
Trypanosome. That would be a protozoan,
Definitely would
wouldn't it be?
Single cell.
Should I look under African?
If you'd like.
If I'd like. I mean, if I'd like. I want to know what's correct.
Yeah, I would look under African.
So we have
Yes.
tropomastigotes invade the bloodstream.
Trypomastigote.
Did I say tropo?
You did. You said tropo.
Wow.
It's all right. You were just testing me.
And you have metacyclic trypanosomes, but no promastigotes. No. You should
Promastigotes
really-
are the ones that you can grow in vitro, by the way. So if you take, for instance, someone who's infected with Leishmania, which is a related parasite, and you take a little scraping from the sore, right, if it happens to be dermal leishmaniasis, and you put it into tissue culture, which is, in this case, an insect tissue culture, which we've discussed in the past in our basic biology of parasites lectures, you will get these amastigotes to transform into something called promastigotes. And the promastigotes are all flagellated, and they're all specific for the insect stage of the infection. So I think all of the Mastigophora, which is the general category for flagellae containing protozoans, that are hemoflagellates, so these are ones found in the blood, they all have promastigote stages in their vectors. So when you say
Right.
promastigote-like, it's based on the length of the body and the width of the body of the single protozoan. And they have stumpy forms and long, slender forms that they identify as being the difference, because if you
Okay.
look at the genetics of this, you can't really tell.
So
them
let's. So far, we have cells that appear to be gametes.
Yes.
Okay.
Yes.
They express meiosis specific genes.
Right.
They're in the salivary gland.
Right.
If you mix them, they fuse.
They do.
Okay. So and they
So-
call these promastigote-like cells.
Yeah,
PL.
because they resemble the
Yeah.
infectious stage from the insect.
Now, why would these have two kinetoplasts?
That's a good question.
That I don't know, because you're going to have two of these fusing, right? So why would you
Yeah.
have two? You're going to end up with four kinetoplasts, right? Do you understand this?
Maybe they shed them after the gametes form, and they obviously revert back to, don't they? Because the trypanosomes that are present in the infected host, let's say a human for sleeping sickness, what is the N number of chromosomes in that organism that circulates throughout the blood?
The trypanosome?
Yeah.
I don't know.
I think it's one.
Well-
It's half. It's half. It's
Well, but hold on a second.
They have one
It's half-
chromosome?
Hold on
I
a
believe
second.
it's
So,
half.
so how many kinetoplasts does each?
one
Okay.
per organism.
That's right. So if now the ones that have fused have two kinetoplasts,
But the fusion
Yes.
of the
Go ahead.
nucleus of the organism is totally unrelated to the fusion or
Yeah,
not
I
of the
understand.
kinetoplast.
right? But
Right.
now you started off with two gametes.
I know what
They've
you're saying.
fused.
I know what you're saying. So
and
they get the
and now
fusion-
and the cells have fused,
right,
and now you have a new cell that has two kinetoplasts,
perhaps,
likely originating from each of the
or it could
originating.
throw one off.
Or you could throw it up, but we haven't gotten there yet.
We haven't gotten there yet, so we have two choices here. One is it could keep them. They could fuse too, though, right? Which would really be confusing if you ever looked at the ultrastructure of the kinetoplast. It's really quite involved. It's even more involved than a regular nucleus, I think, because it's all circles. Total mess, right? Took a real.
Yeah. So we don't understand why there are multiple
We don't.
kinetoplasts.
We have
Okay.
no.
The other
answer.
thing is they find that some of these kinetoplast-like cells have two flagella.
Exactly.
Why would they have two flagella?
Well, because they started out with one, they fused, they kept.
No,
the
but this is
flagellae-
pre-fusion.
The flagellae are attached to the kinetoplast.
Yes,
You're going
that's
to keep
true.
the kinetoplast. You're going to keep
Oh,
the flagella.
so they want to
also.
see if having two kinetoplasts, you always have two flagella.
You
So
do.
most of the time, actually, with two
right.
kinetoplasts, there's only one flagella. So that's the relevance of that.
Right. Yeah. The flagellum doesn't attach to the nucleus; it attaches to the kinetoplast.
But I guess one of these kinetoplasts goes away at some point.
Apparently,
They don't actually talk about this.
they don't.
You know,
They don't.
I would like to have known that.
Right, because the end result is
These are going to fuse, right?
right.
And then become an adult trypanosome. An adult trypanosome. They're going to fuse and become. They're going to
a
have.
child.
A child. That's
And
Not
they
an
will-
adult just yet.
That's right. That's right.
They will be released from the salivary gland and
They
go
will.
into a host and mature, right?
And then what happens?
What do you mean? Then what happens?
Well, then they have to reproduce like crazy in order to make a lot of them. So then when
Yeah.
the tsetse fly bites, you get to initiate the
Yeah,
infection. So my big question then, I guess, if I didn't know this paper or didn't know much about trypanosomes, is are the infectious stages of the trypanosomes 1N or 2N?
The infectious stages are called what? What's the name of those?
They are promastigotes.
Yeah.
And the answer is
No. Are we going to find out by reading this paper?
No,
That's
you're
what
not
I'm
going to
hoping.
find out. Well, actually, they do look at DNA content.
They do.
They do that by staining with dyes that stain DNA. They use two different ones, DAPI and propidium iodide.
Right.
And then they measure fluorescence.
Right.
So by measuring the intensity, you can get an idea of whether it's 1N or 2N.
Exactly.
Right. N being a chromosome, right?
Right.
Chromosome number.
Yep.
So the 2K1N, which is two kinetoplasts, one nucleus, and the 1K1N PL cells were haploid
Haploid.
relative to metacyclics, which is diploid.
Okay.
Now, what's a metacyclic?
It's the stage that usually occurs in the gut tract of the vector. I'm only familiar with the metacyclics that occur in sandflies and in reduvid bugs. So I'm not very familiar with the African version here.
They say that the metacyclids are present. It's the mammal infective stage that's present in the salivary gland,
Okay.
and those are diploid.
So the procyclic develops into the metacyclic, which then infects the mammal.
Right.
So
So the
the trypanosomes that circulate throughout the blood of infected hosts in Africa are diploid. That's what this paper says.
That's correct. The infective stage is diploid, and these promastigote-like cells are haploid, according to this DNA analysis.
Okay.
Okay.
Which allows for mating to occur. Otherwise, it just to keep accumulating
well, yeah,
chromosomes,
of course.
right?
You have to have a reduction, which is
to get
meiosis.
and then a division. That's right. Okay.
Yeah, that's basically the paper.
So that summarizes something that I didn't know before, obviously, but that didn't mean much because I'm not closely following this field as I did, let's say, for Trichinella, the organism that I worked on. I'm intrigued to learn that this organism, once ingested by the tsetse fly as a diploid organism, then undergoes a division cycle which creates a haploid organism which can then mate again. Now, how does that actually result in a recombination event that has an evolutionary significance?
Well, it has to have.
But it's a clone to begin with. Maybe they took the clone right out of that animal. I don't think tsetse flies feed multiply and then all of those trypanosomes all mix together at the same time and then exchange genes. That's not what I think happens.
Well, but
one blood meal is enough for the trypanosome for the tsetse fly to reproduce.
Yeah, but a single blood meal may have multiple different trypanosome organisms, and so
It's true because animals don't die from this, right? Only humans and certain European cattle die from this infection. So the wild animals, like lions and tigers and bears, oh my, all of those animals would be able to accumulate multiple infections over time, or do you think the innate immune system might prevent that from happening?
But that's different.
Well, then you get...
The question is whether in the fly midgut. So you made a really good point. One possibility is that a tsetse fly takes a blood meal and is infected with a single organism, a single trypanosome,
that would be interesting.
or even multiple trypanosomes, but they all came from one infected host, and so those organisms are all genetically identical.
Right.
They're all diploid. They become haploid.
Yep.
And then suddenly in the salivary gland, they come back to being diploid.
Exactly.
So what have you gained?
Yeah,
exactly.
Right.
Zero, right?
It's a zero-sum game.
Right.
The other explanation is that when you take a blood meal, there are multiple different trypanosomes that are taken up.
Right.
Or if you take multiple blood meals with from different hosts, like was the example with Rhodesiense and picking up genes from what? Remind me.
They didn't say.
Did they say where they came from?
They didn't say.
They didn't say where they came from. No, they
didn't say. No.
And so that's an example where a tsetse fly perhaps would have been infected with two different.
Right.
Yeah.
I mean, there are other trypanosomes out there too. I mean, there are Evans eye that infects horses and stuff like this. So maybe they would infect zebras, and maybe they could pick them up like this.
Well, now that they know there is possibly or probably sex, they could look for it. Maybe they didn't look before.
They could. What really hasn't happened, though, is for the resistance or the susceptibility to that host serum protein that prevents the organism from dividing has not crossed over from West Africa to East Africa. It hasn't gone that way, even though And it's interesting to look at the distribution of the vectors. The tsetse fly isn't just one species; it's a whole bunch of species, and there's something called the savanna tsetse flies on the in East Africa that live like 1 per mile. That's the density of this trypanosome vector. If you go to West Africa, they're called riverine, and they all breed along the rivers, and there are millions of them along the rivers. So it's very easy to pick up trypanosomiasis in West Africa, and it's very difficult to pick it up in East Africa based on the density of the vector itself.
Okay.
So that has ramifications for exchanges and stuff like that too, I guess. Because I can't imagine that the tsetse flies haven't blown over sometime with a storm or a bird that transports one and then gets loose in another place. Sure. But if a West African trypanosome gets loose in East Africa, there's no water, so they would probably just die there. because they're used to breeding next to rivers, and there are very few rivers in East Africa.
So you're saying the practical consequence of this is maybe minimal?
Yeah, I mean, I'm just speculating here, but I'm based on my knowledge of listening
Yeah.
to people involved in these vectors for a long time. You start to think like those people after a while, and I've heard these conversations a lot. And I'm just reiterating, maybe some very old literature right now. Maybe other people out there that are listening know more about this than
I don't know.
But,
I doubt it. Well, but we do have parasitologists listening, so
we do.
They,
And,
by definition,
I hope these guys are listening in Bristol and
I'm sure
Cambridge.
they're not.
Because, no, I would like them to though, because they could probably add to the conversation through email.
Well, we could send them the link and say, "Can you please answer
Let's
our
do
questions?"
that. No, we should do that.
All right. So their conclusion is.
Yes.
We conclude that haploid PL cells are trypanosome gametes, although formal proof will require demonstration of exchange of nuclear and kinetoplast DNA.
Nuclear and kinetoplast.
Exchange between the two fusing gametes. Well, that becomes one cell, so what do they mean by
Right.
exchange? Do you
Right,
understand
exactly
that, Sagi? Exchange?
Well, when you have this is the principle of sexual reproduction, right?
Right.
You
You do exchange. That's
Yeah.
right. Exchange is the pleasant part of this whole thing. A fusion, perhaps, of or confusion in this case.
Well, in this case, they have to show they have two differently marked PL-like cells, the gametes, and show that in the fused cell, those markers are together, right? Both for nuclear and kinetoplast DNA.
Yeah, well, you said you can transfect very nicely, so you could actually do these experiments and
Transform.
come out with a very nice result.
You should really use
Now,
transform.
Yeah, you should.
The thing for me that seems most interesting, and not just this, but as a just fundamentally, how do you evolve the ability to sexually reproduce? How do you evolve the ability to recognize that another trypanosome is one of your brothers, sisters, is one of your own, rather than fusing with
Sure.
a cell from
Yeah.
the tsetse fly?
Exactly.
And that's a non-trivial problem that had to be solved
Yeah, it's
at
a
some
good
point.
I think ICAMs and
Um,
ECAMs and all those cams are involved in this.
Maybe.
Maybe there are probably lots of things, but this is
Yeah.
clearly there's very strong selection pressure
Sure.
to increase genetic diversity,
Yeah.
and so
Yeah.
once it happens, there's very strong positive reinforcement for it.
Right.
But how do you cross over that hurdle where you know that that this other cell is benefit?
Right.
It's a good question.
Right.
And in fact, along that line, they say the trypanosomes are among the earliest diverging
did say
eukaryotes, right?
Yes.
Long ago.
That's right.
So this means that meiosis and sexual reproduction are ancestral and ubiquitous features of eukaryotes.
That's incredible because the most ancient one that I know of is Giardia.
They do sex, right?
Well, they say they have the meiosis genes.
It's
but
true.
They didn't say they've actually seen the sexual stages of Giardia.
Yeah.
And that's a bizarre parasite because it has two complete nuclei. No kinetoplast, but it has complete nuclei. And both nuclei are active.
Beautiful,
They're both
beautiful looking parasites.
And then they're gorgeous.
Giardia are beautiful?
Yeah.
They're gorgeous.
Really?
They're
They look like they're
absolutely
looking at you.
wonderful. This is
They
true.
have-
We call
they
them
have two
monkey
nuclei.
faces
Two
or
nuclei,
stuff
yeah.
like that. But
All right, let's go through some of the parasites. Leishmania, sex,
sure, it's an apicomplex. No, it's not
and
It's a
trypanosome.
hemoflagellate.
Plasmodium for sure.
Absolutely.
How
Crypto.
about the Babesia nigraria, acanthamoeba, blastocystis,
We've been through toxoplasma.
Wait. You have to do it by groups. You have to do this by groups first.
protozoa of
So
minor medical
hemoflagellates, we think yes.
Okay,
Apicomplexa,
those are the malarias.
well, and the cryptosporidiums and the cyclospores and those work,
They
and
do.
the toxoplasmas, we think yes.
Okay. What about nematodes for sure?
No. We go back to the amoebae first. Now, do
you want-
amoebae have sex? We don't think so. You know why? Because they're polyploid. Most of them are polyploid.
But they are ancient eukaryotes, aren't they? Are they
They're-
before the
they're
trypanosomes?
even- well,
They must be,
It's
no?
hard to know that because if you look at the tree of life, as we now know it, systems biology, what do you think?
Oh, it says here,
Are
Dickson,
the-
I just Googled, do amoeba have sex? And I found an article in Live Science saying researchers are rethinking the asexuality of amoebas. They now have evidence of amoeboid sex lives.
Really?
Amazing.
That's incredible.
Yep. University of Massachusetts.
They have evidence, but do they have definitive proof?
Yeah, well, I haven't, of course, looked at the original paper. But
Right.
there you go.
Right.
An amoeba might have sex.
So are they phylogenetically ahead of or behind the most
Trypanosomes.
primitive mastigophorans?
I don't know.
How does that play out in terms of the phylogenetics? I always
don't
thought
know.
that the hemoflagellates, or the flagellates rather, right, were the most primitive forms of protozoans identified. Because obviously Giardia is on that list, and that's way... If you look at the connection between the eukaryotic and the prokaryotic and the two forms, the archaebacteria and the bacterial lineages, it almost fuses with that little Y-shaped junction for genetic analysis. But there must be something before that, right? That preceded them but still is a eukaryotic. And maybe those ancestors are gone, or maybe we haven't found them yet. Maybe they're sitting at the bottom of the Marianas Trench waiting for us. Which is like, which is the guy that went down by himself? What a frightening trip that must have been. The guy who filmed, who made the movie Avatar. Blanking on his name.
Cameron.
That's right, Cameron. He went down in a submersible down in
No, he did, didn't he? By himself, yeah.
by
Really?
himself. That he built. He built this thing by himself, and he went all the way down to 36,000
Why are we talking
feet.
about this?
Why? Because that's maybe where the most primitive eukaryotic cell might lie.
It says here they're talking to an author of this article on amoebal sex. The last common ancestor of all living eukaryotes had to be sexual.
Ooh.
Sex is the rule and not the exception. That's
Well,
what he's
bacteria
saying.
exchange genetic material. You wouldn't call it
Not
sex
bacteria. These are
necessarily.
eukaryotes.
They're- No. But bacteria precede eukaryotes, so you've got conjugation between bacteria, right? You've got exchanges of genetics.
Yeah,
So,
but
and
that's
where they-
just exchange. Here, sex is when you make a
Understood.
gamete and then you fuse them.
No, I got it. I
You know,
got it.
bacteria do. It's just slightly different. It may be.
No. I totally
We
agree.
call it sex, but it's not really.
So if you went back through phylogenetics again, or phylogenomes, how do these three meiosis genes play out in terms of prevalence?
That's a good question. I don't know the answer to that.
I would love to know that, and that's a great systems
If they're
biology
conserved.
question.
I
that we
presume they're conserved because he said they did.
could- yeah, but conserved
she
among
said-
who? How far back do can you find any of examples of these genes in prokaryotes?
Well, the two papers. One of them is a phylogenomic inventory of meiotic genes,
Yeah.
evidence for sex in Giardia, and an early eukaryotic origin of meiosis.
Yeah.
So that's as far back as they could look, I suppose.
Right.
Okay.
Yes. But I also want to say something about, you asked about worms.
Yeah, you didn't answer me.
Well, I'm going to answer you.
Do they have sex?
Of course they
I know
do.
earthworms do.
No, that's right. No, all the parasitic nematodes.
Cenorhabditis?
Sure.
Okay. Those are the
course.
nematodes. What about
cestodes. Here's a bizarre group. All right?
Each segment has sex,
They
right?
have sex, okay, but they have sex with themselves.
Well, so
It's a clone. This is a clonal organism. You eat one
segment.
cysticercus,
Yeah.
right? And you eat that, and it develops into an adult tapeworm in your gut tract, and it produces all these segments, and these segments are busy mating with each other. That's the same
pretty
genome.
bizarre.
That's the same
That
genome.
is
Now,
bizarre.
But now
maybe-
they mate with only the segment that's next to
yeah,
them.
that's right. They mate with itself. It mates with itself. But it's a colony. It's not a single organism. It's a colony of similar organisms, or maybe identical organisms. And each segment is at a different stage of development. Everything is bizarrely made.
All right, so that would
What would be
include-
the advantage of a tapeworm having sex, though? Why don't they just produce segments?
Must be an advantage.
With
otherwise
haploid eggs or
wouldn't be there,
something.
Dickson, right?
It's got to be.
Taenius, all those tapeworms, right? And
gotta
then
be.
Diphyllobothrium.
Those are all cestodes, and all cestodes have sex.
Cysticercosis.
All
All right, so they have sex. And what about
yeah,
trematodes,
definitely.
schistosomes,
There are two kinds. There are
etc.?
male and female trematodes, and
Okay.
there are hermaphroditic trematodes. But the hermaphroditic trematodes don't mate with themselves. They have to mate with another partner. So they, in essence, they do a. You're going to have to eliminate this one too.
No NSFW.
What?
No NSFW.
Right. I'm not going to do that. But they line up in a certain way so that each can exchange materials with them.
Head to tail.
That's what
Head to tail.
That's
Thank
what
you.
earthworms do.
That's what earthworms do too.
Yeah. I remember hearing about that in biology class.
Sure. And you know the sex organ of the earthworm is called?
No.
It's called the clitellum.
Clitellum?
The clitellum.
Wow. All right. That's a good paper, Dickson. Thank you for
Oh, no.
picking
It
I like
allowed
it.
us to pull into the conversation Sagi, which is a great introduction to podcasting in general.
Do you ever been on a podcast, Sagi?
No, this is my first.
What do you think?
True.
Can they be fun?
They can with the right.
You could just
with the
have,
right,
not
with
with
the-
this
in
one.
the right
No, I know what you're
company.
saying.
You gotta have the right company.
With the right people in the room, it could be very interesting.
And given when people maintain appropriate distance from their microphone.
What you can't do is you're not allowed to allow your ego to get in the way of this conversation, because when that happens, then it becomes a very negative.
Do you have an ego, Dickson?
Kind of. Yeah, I do, but I don't bring it into this room. I make sure that it stays out over there because, I even got where Norwalk virus came from wrong.
Yeah.
To begin with, in our original podcast for This Week in Virology, I readily admit to my mistakes. Readily. And I think if you do that, then you can maintain balance in your life. And if you hold dear to the mistakes that you've made and take offense at them, then you'll never be good at anything else. So that was a lesson I was taught early on.
The good thing about this paper is it's open access.
It is open access.
Current biology is open, so
It
everybody
is.
can go see it and the nice pictures. So let's read some email, Dickson.
I'm going to do that right now.
First one is from Tim.
We'll have to get back to the email first.
Just do that without talking so I can read email, okay? Tim writes, I listened to the latest TWiP this morning. Dickson mentioned the herbicide atrazine, but thought it was a fungicide. It's actually a herbicide in the photosynthesis inhibitor class. Another bit of trivia about ag chemicals is that old chemicals like atrazine are making a big comeback in usage due to resistance problems emerging in the field. Too many farmers were spraying nothing but glyphosate on fields due to that being the first GM herbicide resistance trait, and now weeds have caught up. On the subject of monarchs having a bad taste, so birds leave them alone, you seem dubious of bird memory and the difference one bird's experience meant for monarchs on the whole. I think the reason it works out well as defense for monarchs is that the generation interval for insects is much quicker than birds, and some birds live quite long lives and would most likely experience the taste of monarch early in life and not forget, because they are not only bad tasting, but I believe poisonous due to milkweed that they feed on as larvae. I've tasted milkweed, and if monarchs taste like that, I can see why birds would avoid them. That was my comment. I couldn't understand how, the memory of a bird eating a monarch would, perpetuate throughout the colony of birds or whatever.
Well, there's a ton of monarchs, but there are very few viceroy butterflies.
Yeah.
And so the monarchs taste bad. The viceroys don't, but the viceroys take advantage of the fact that monarchs taste bad to protect them
so
from
every
being
um, bird
eaten.
has to taste a monarch
That's
to
easy
make this work.
because there's tons of monarchs.
Okay.
Well, but that rats
Uh-oh.
have, and I don't know much about this, but rats do have a way of informing the rest of the colony when food is good or bad.
Huh.
Now, maybe rats are more clever than birds, but
Okay, that could be also.
that might be a way to.
Maybe there is evidence. Okay. I found it interesting you have the view of humans as separate from the general ecology of the Earth. Full disclosure, I wrote a paper comparing humans to weeds and pathogenic bacteria in college, but have since changed my mind on that subject. I've been working on changing people's minds about that on social media because it seems once we view ourselves as separate from the rest of the world we live in, it seems to give people license to do things that make no sense from an ecological sustainability standpoint. To put another way, stealing a term from economics, there's a risk of moral hazard when one divorces themselves from their surroundings. I'm not saying this to criticize your thoughts on it, and I'm sure you have a much more nuanced, full view of the subject. Just some thoughts of mine on that topic.
Those are great comments.
So that was my feeling. If you took humans away from Earth,
right.
probably there wouldn't be a big impact like any other. I mean, yeah, the things we kill would grow more and all that stuff, but it would be good for the Earth for us to get out of here. It would return to its natural state.
That's what
right?
Alan Weissman
There would
writes
there would
about.
certainly be changes because
Sure.
our activities would no longer exist. So someone said, "Oh, the deer would overgrow." Yeah, but then
No, but
whatever
then the
eats-
predators would come
the
back to eat
predators
them.
would take care
That's
of them.
right, that's
The
right.
wolves that are in
That's right.
small number in New Jersey, the foxes, the coyotes, they would
That's right.
grow. You know,
That's
that you'd
correct.
have a balance.
That's correct.
So-
No, I'm not sure that we actually did say that we separated humans from them. We just said, what would happen if human beings disappeared tomorrow?
Yeah, well, I said there'd be no impact, and that's sort of separating them
well,
from the ecology. So there would be an impact, but not because it's only because the stuff we're doing is negative. I think. So all the forests we clear, those would return.
Sure.
Right. The atmosphere would change as a consequence. Probably warming would subside because we're not pumping CO2 into the
Yep.
atmosphere and so
Yep,
forth.
there are people that wouldn't believe that, but I believe that's true.
So whereas if you took the trout out of a food web, that would have a big impact.
Yeah, well,
right?
you're talking about keystone species. That's the definition of
Yeah,
a keystone species. So the point is that there's a book on this subject. It's called The World Without Us.
Yeah, right.
And that's
You mentioned that
a wonderful
once.
book to just speculate. What would the Earth look like one day after that happened, 10 days after that happened, 100 days, and 1,000 days? What would the Earth look like after human populations completely disappeared? And it's an interesting speculation rather than
Who wrote that book?
Alan Weissman.
And who is he?
The publisher was...
Who's Alan Weissman, he asks you?
He's a person who got this idea in his head to write this book,
Just
I
a-
guess.
it's
I
just
don't
a
know his
writer. Just a
background.
guy.
I don't know his background.
He's just a guy.
Just a guy.
He's just a guy. He made a
All right.
pretty good profit on the book, I must say, and it was published by St. Martin's Press.
Tim, who wrote this letter,
Yes.
is a farmer in Minnesota.
Right.
He writes to us often. He raises
Right.
cows and probably other stuff too.
Right.
And he just started a podcast.
Cool.
I have to get a link to it and listen to it. Check it out.
Excellent.
Yeah, Dickson.
Yes, I'll read the next one.
No, you can't read any email. Go ahead. Please stay near the microphone.
I will when I'm...
When you're what?
Nothing. I'm going to do this right now. So, Scott writes, Hi, Vincent and Dickson. I just finished episode 64 and wanted to drop you guys a note to hopefully fill up your inbox since you only had 4 emails. I am a hydrogeologist working for an environmental consulting company in Cincinnati, Ohio, which is currently overcast, 24 degrees Fahrenheit, 79% humidity, winds at 8 miles out of the south-southeast.
Sounds like Manhattan.
That's right. Though hydrogeology has almost nothing to do with biology, I love listening to TWiP. Oh, I wouldn't say it has nothing to do with biology. I think it studies the places where water can be, and that's
You're
without
telling
water,
a guy
you
who
die.
works in the field?
Without water, you die.
You're telling a guy who works in the field?
No,
what
but
it's
he
about?
knows this, so I'm saying that you're diminishing the importance of your subject area, I think. He says, I don't have almost nothing to do with biology, and I love listening to TWiP, TWiV, and TWIM while doing my mundane data manipulation, and I am a lover of anything science. The furthest we tend to dive into biology would be the injection into groundwater for dihalococoides for the sequential reductive dechlorination through the daughter products cis dichloroethylene and vinyl chloride to ethylene.
Ethene.
Ethene. I'm sorry, ethylene.
Not ethylene.
I said
Ethene.
ethene. It's hard. That's right. Guess it's a little more relevant to TWIM than to TWiP. Please keep going off topic. That's impossible not to. I love hearing the banter back and forth between you guys. Anyway, I love the podcast and look forward to every episode. Hopefully there are many more to come.
Well, that's a very nice email.
That was a great email.
Thank you, Scott. Next one is from John. Hello, TWiP.
Yeah.
I just finished listening to episode 67 in which Vincent asks, why was a priest doing experiments?
Uh-oh.
I just didn't know that.
This must have been about Mendel.
Oh, it was. We did mention
Is this
Mendel.
about Mendel?
We did mention
Well, you
Mendel.
said Mendel did experiments. Yeah.
I did.
I'm a parasitologist who will be ordained a priest this June and will teach biology at Creighton University.
Right.
I earned my PhD in evolutionary biology from OSU before entering the Society of Jesus a.k.a. Jesuits, in 2003.
Right.
After 10 years of studying philosophy and theology, teaching biology, and doing research, I could probably devote an entire TWiP podcast to the topics of priests doing experiments, as well as parasitology and religion. But let me briefly state the following. Christianity helped develop science because it assumes, unlike some Eastern philosophies, that the world is real and not an illusion. Christianity also assumes that the world of creation remains distinct from the world of the supernatural divine. So unlike many pagan religions that believe in nature gods, Christians assume that nature follows laws that can be studied. Finally, Christians believe that God created the world good. Thus, creation reflects the Creator, and so learning about nature teaches us something about God. In other words, investigating the world of nature and is worth doing. You and your listeners can check out some of my online articles on this and related topics at thejesuitpost.org. Okay? Thanks for the great podcasts, and keep up the good work. I plan to use some of these podcasts when teaching parasitology. Peace. So I understand that priests can do research. So actually, we should have John here on an episode of TWiP so he can talk about this, because a lot of people are anti-evolution because of their religion. So I don't understand how you can.
How much are they anti-evolution?
All right, what are they?
I think it's probably a little bit more complex than that.
I tend to be a reductionist. Yes.
I mean, it's many who are very religious.
Yes.
Don't necessarily deny evolution, but that perhaps that it's a process that was started by a divine power.
Right.
And so they're not necessarily mutually exclusive. I would love to hear what who was this? John has to say about it. But
I do too. I think we should have him on. You can join us for that episode.
Right.
Would you like to do that?
Yeah.
Well, Stephen Hawking, we mentioned him before. Stephen Hawking actually had an official show on the Discovery Channel entitled "Why There Is No God."
Yeah.
And he used the following argument. He said that if the entire universe began as a singularity and then developed into what we have today, then if you go back to the singularity and say what was the characteristic of the singularity, then it's infinite gravity. Okay? And infinite gravity means that there is no time, because we know that time and gravity are linked. So he said, if you go back to the singularity when there was infinite gravity, that means that there is zero time.
If Stephen Hawking is listening, I believe.
And if there's no time,
this is a formal invitation.
Right. And if there's no time, then there can't be a before. And if there's no before, then that means that there can't be a God, because there was no before, because there was no time. That was his thread of the argument.
Okay.
That was the thread of the argument. It was an interesting argument, but I still would challenge Stephen Hawking to account for the fact that if the singularity was infinite gravity and stable, what caused it to destabilize? Or was it only a mathematical point at which it existed, and before that, it raises huge.
It's interesting you mentioned the singularity, because in the car the other day, my younger son said, "Dad, could we deal with the God evolution issue in this way? Let's say that God did the singularity." He was just playing around, and that happened, and that was his doing. But then everything else was
playing
evolution.
around.
I said, I don't think you can just restrict him to one thing like that. But it was interesting that he came up with that. He said he had a discussion in school. Dickson, can you take the next one?
Yes, I can.
No, you can't go away from it. Oh, I see why you did that.
I did it as a joke.
Isn't it perfect
Thank you, Vince.
that
Thank you, Vince.
we came to this one now?
So here's it's very, very appropriate. Alex writes, "Hi, both Vincent and Dick. Love the podcasts in every way but one thing."
He's doing it on
Vincent
purpose.
is very clear to hear. Dick fades in and out, meaning a volume can never be perfectly set. Although I'd illustrate below. Please fix this so I can continue to enjoy stories from the world of parasitism. Thanks. Well, we've fixed it. I guess I do tend to bob up and down. Maybe Ali.
You move back and forth.
Colin. All right, I'll stay a set distance away from the microphone forever from now on.
I could tell you forever, and you wouldn't listen. But now that we have a listener, and the listener sent a nice graph.
No, I'm
which
totally
shows your volume going
apologetic, and I'm sorry that I've.
I'm going to use that as the episode art. What do you think of that, Dickson?
Whatever you'd like.
You don't care, huh?
You're sure I care? Of course I care.
Just stay near the mic.
I'm attempting to do that.
It's called mic technique. I took away the boom because it didn't sound very good.
That's true.
But now you have to be a little more conscious.
I'm trying.
The next one is from Christophe, who writes, "Hi, just thought I would pass on this link in case you hadn't heard about it. I have not watched this documentary series, but I think I will try and track it down. I heard about it on the BBC Science Hour podcast today and would like to hear Dickson and your thoughts about it. P.S. I love the TWiP Twin podcast. I'm only a high school graduate and self-taught web designer, but find all the TWiP fascinating. Keep up the great work." Now, Dickson, there's an article here.
What was the link to?
Oh, it's a very long link. I can't...
What type of documentaries?
Well, it's swallowing 3 cysts which are twitching with tapeworm larvae. Sounds like the stuff of nightmares. But for the past 2 months, BBC presenter Michael Mosley has been taking part in a toe-curling experiment, allowing 3 worms to live and grow in his gut. As part of BBC4's Natural World season, Dr. Mosley infected himself with some of the world's most prevalent parasite, including leeches, lice, and mosquitoes.
Right.
He infected himself with a mosquito, right?
Well.
So what is this worm that he's eating there, Dickson?
Well, that would be a cestoid, wouldn't it?
Did you look at it?
Not yet, but if he mentions the fact that he's eating 3 cysts, which are twitching with tapeworm larvae.
This is what he's eating. Look.
No, he didn't eat that.
What is that? It's Ascaris, isn't
That's
it?
the tapeworm. No, it's not. It's a tapeworm. It's an adult tapeworm. If you swallowed the segments of an adult tapeworm, you wouldn't get infected.
Yeah, see, Sagi knows you're moving away.
So I'm sorry. I'm trying to read and take
swallowing
part in
3 cysts, which are twitching with tapeworm larvae.
Cysts,
yeah.
That's different. Those are cysticerci. That's what they're known as. The cysticercus stage of the tapeworm. So if you eat that stage, you get the adult. That is the larval stage of the tapeworm.
He also swallowed a pill camera, which gave him a gut's eye view of the parasites.
Right. So if that's a beef tapeworm, I mean, I would, I know someone who actually worked on the segments of the tapeworms to study the active transport mechanisms that the tapeworm undergoes in order to get its nutrients. Mm-hmm. And in order to do that, of course, he had to have readily available tapeworm segments. So guess where they came from?
Beef?
No, they came from his gut tract.
Now, Dickson, correct me if I'm wrong, but I seem to remember stories from professors that I had, both as a master's student, also studying parasitology, and as a grad student.
Go on.
Stories where parasitologists working in the United States would essentially import some of the parasites they were working on by infecting themselves.
This is correct. And one of them died from that, as a matter of fact.
And this was not limited to
tapeworms.
To
no,
tapeworms.
That's right.
This was
there's a book,
sort of a common practice.
Listen to this. There's a book called Guinea Pig Doctors. It was written by 2 Baltimore Sun reporters, and it was about all of the experiments that physicians and people in medicine have carried out on themselves because they couldn't get past the IRB, or they didn't have an IRB at those days. One of them was this case in point where someone who wanted to work on schistosomes in this country, and we didn't have them here, infected themselves with schistosome. I believe it was hematobium, but large amount, unfortunately, because it ended up being a fatal case, and brought it back to this country, only later on to discover through someone else's research that you could have other hosts have this infection and work on it that way. So he made a fatal mistake in 2 directions at once. This guy, tapeworms don't really do much. Okay?
So what would happen if he swallowed these cysts?
They would develop into adult tapeworms.
Okay.
So what he's showing here are some of the segments from his own tapeworm.
All right, now.
So he swallows this camera to get a picture of his adult tapeworm.
All right, what he said here, Dickson, is that he put on 1 kilogram in weight, and he said the worms made him eat more. I definitely wanted chocolate and carbohydrates. Is that true?
I think that's psychological and not physiological. I I wouldn't buy that. No, I would doubt that. I would tend to doubt that.
The evidence is certainly the opposite.
It's quite
that people lose weight. That people lose.
Well, no, that's also untrue. I mean, we've gone through this before. These tapeworms, they don't absorb much from your gut tract, to be honest. They're very, very low metabolic rates. They're anaerobes, basically. So they don't really carry out much in terms of aerobic metabolism. They're probably microaerophilics.
So he has this worm in him now, right?
Yeah.
What's going to happen to it?
They could treat it with praziquantel and get
Otherwise, if he leaves it there, what happens?
nothing.
It will just coexist with him?
Sure.
It won't obstruct his
No,
gut?
the Chandler.
He has a single worm in him, right?
No, I know that. So there was this famous textbook called, I believe it was called Basic Parasitology by Chandler and Reed. Clark Reed, I knew. I didn't know Asa Chandler. But Asa Chandler was a tapeworm person in Texas, University of Texas, I believe, who actually had an adult tapeworm and used his own tapeworm segments to do experiments on.
So in this article, he just talks about the life of this tapeworm,
Yeah.
its natural host. So it's a way of him teaching people, which is fine, but we do it here.
Hands-on. They would call this hands-on
No, but Dickson,
parasitology, but...
Do you really need to say, you can't see anything? Do you need a guy sitting there saying, I swallowed it and this is what happened?
No,
How about the TWiP approach where we talk about it back and forth? Isn't that just as
I
good?
like this approach much better, especially if you were talking about Trypanosoma brucei, for instance.
Yeah, you can't use
disease.
pathogenic things.
I
Anyway,
wouldn't do that.
Christophe, listen to our segment on tapeworms. I think you'll
No
enjoy
pun
it.
intended.
Segment, yes. All right,
Good.
so...
Incidentally, speaking of BBC documentaries,
yes.
If listeners, and I'm from these emails, clearly they're all very clever and scientifically savvy, so they must know who David Attenborough is.
Oh, yeah, sure.
And there is a new series that has come out recently on Galapagos. And which is very fitting also for what the topic is today, speciation.
Absolutely.
That's right.
Fantastic.
Nice.
Really, really fantastic
Oh,
series.
I look forward to seeing it.
Yeah,
I
they're very
love
good.
Attenborough.
Very, very, very
He's
good.
fantastic.
Dixon, the next gentleman, Robin, sent a link to an article
Yes.
called Arctic Whales Infected with Domestic Cat Parasite. It's for you too, Sagi. Toxoplasma
right.
gondii has been identified in beluga whales in the western Arctic.
How about that?
Yeah.
How about that?
It's amazing. Toxa has been found, I think, in every vertebrate that has been looked at.
It's the most successful parasite on Earth.
Says here for the
Dolphins.
for the Inuit people who
And
eat
also
the whale.
kills otters, right? Kills sea otters.
For the Inuits who eat the whale meat,
Yeah.
this has prompted a health advisory. Would you agree with that?
I would, because remember way back in the Trichinella description for that podcast that we had, we talked about Eskimos, and we talked about what the word Eskimo means in their own language. And remember what it meant? It means raw meat eater.
Mm-hmm.
So being a raw meat eater, what would they cook with? Of course, because there's nothing in the Arctic, although things are changing. We could have trees up there soon, but there's no fossil fuels up there to use, so they eat their food mostly raw. So that's why this is a big
they
problem.
suspect that there's been an influx of cats to the Arctic,
Sure.
and they've imported it, and their feces get in the water
Yep.
and washed into the ocean. And
That's
they say
right.
with warmer temperatures in the Arctic now, water is
Yeah,
mobile for
That's
longer
right.
periods.
This is exactly
Global
right.
warming, eh?
That's true. That's true.
But now, toxoplasma infection is not necessarily problematic for people unless they're immunocompromised.
Correct. No, this
Or
is not a
become
human problem
immunocompromised.
necessarily, but it's
Pregnant,
knocked
right?
off.
Or for pregnant women.
It's
or
killed off a bunch
actually
of-
not pregnant women. If a woman has never been
Right.
infected before
Right.
and becomes infected during the first or second trimester.
That's right. That's what's a big problem.
But
Congenital toxoplasma. That's
yeah.
right.
The next one is also about toxo. Dixon, it's from Marianne. Please read it.
I would be happy to do so. Dear Doctors Racaniello and Despommier, I do enjoy your podcast, as I do all the TWiCs. I have written to TWiV previously, but this is my first opportunity to email TWiP. Your request for emails in this last episode sealed the deal.
It worked, Dixon.
A couple of episodes ago, you bemoaned the lack of ecological-based parasitism research. I smiled to myself because my first undergraduate research experience was in this field. I was a summer student on a field project trapping the prey species of the pine marten of Newfoundland. and studying their parasites. I learned a lot that summer, including that I was not cut out for field work. 2 months in a cabin on the east coast of Newfoundland was enough for me. I happily started my career in the lab studying immunology, virology, cancer, and now vaccines. More recently, I've been thinking more about parasites due to my increased interaction with rescued cats. After losing 2 older cats, we decided to adopt a kitten from a local rescue that helps trap, spay, neuter, and then adopt or release feral cats. Before we could adopt our chosen kitty, he had to clear a pretty significant coccidia infection. Of course, through my renewed interest in this subject through listening to TWiP, I was interested in what type of infection our little Felix had, but learned that most vets only look for the presence of oocytes in the stool, and since the treatment is basically the same for all coccidia, they don't differentiate. We then fostered a stray mother cat who gave birth to 5 kittens about 12 hours after we brought her inside. Those kitties were happy and healthy until 2 of them went to a local pet store who helped out with adoptions. They both became quite ill with coccidia, while the 2 that we kept in our house remained healthy. I suspect that the well-meaning pet store was unwittingly spreading some of these pesky parasites to young kittens through previous inhabitants. Luckily, after several antibiotic courses, all are happy and healthy and in their adopted homes. On a related note, just last week I saw this story regarding Toxoplasma gondii cropping up in a beluga whale in the Beaufort Sea. Well, that was the same story as before, right?
Yep.
The story also mentioned that Sarcocystis pinnipedi, pinnipeds, by the way, are seals, may be responsible for a large die-off of gray seals off Cape Breton in 2012. They are suggesting that the parasites are being released by ice thaw. What do you think? Oh, and as for the obligatory weather report, it's a balmy 4 degrees centigrade here in Halifax, Nova Scotia, Canada. However, we are expecting a mix of rain, freezing rain, and snow overnight, and they are anticipating the temperature to dip to minus 7 degrees C, wind chill of minus 14 C by morning. Still a bit of winter left out here in Canada. Cheers.
What do you think of these Sarcocystis pinnipedi being released by thawing? Does that make sense?
Yeah, sure, because seals come up on the shore and they deposit feces, of course, and things of this sort. And remember, there are other organisms that can carry this as well. So sure, it does make a difference.
So in 2012, there was a die-off of 400 gray seals, and the deaths were due to Sarcocystis pinnipedi that originated in the Arctic.
Look at that. Yep. Very short food chains in the Arctic. So that's the reason why you have these events that you wouldn't ordinarily have, let's say, in temperate zones or even subtropical or tropical
What do you mean
zones.
by that, Dickson?
I mean that there are very few life forms in these areas, and therefore food tends to concentrate up through bioaccumulation and other things too, contaminants in the environment, parasites, etc. Because the number of species that you have to go through in order to get them into the top predators is very short.
Okay. So you think it's reasonable that this
Yeah.
that thawing would explain this?
It would.
Why would it be frozen up there to begin with? From other pinnipeds that
Of course.
excreted it?
Yeah.
Yes.
Yeah. So what ultimately would we perhaps be worried about if...
Turn the mic. You're moving away as you get excited.
No, I'm not moving away. I'm keeping the same
what
distance.
What else should we be worried about?
Well, woolly mammoths were in the...
Viruses. We now know that there are infectious viruses of amoeba in
There was
Siberia.
a big Mimi virus discovered.
It's not
right?
Mimi virus. It's a new virus.
Mimi.
It's not even related to Mimi.
No.
It didn't. It looked like one.
No, looked like
was-
Pandora.
I meant Pandora. It was from
Pandora.
an amoeba, though. We think yes.
Well, they cultured it in amoeba, but
Right.
we don't know what the natural host is.
Okay.
So there's probably a lot of stuff frozen up there that's going to get released as it
Right,
thaws, right?
exactly
Someone should make a movie about that. All right, let's do one more. This is from Oscar with a K,
Do it.
who writes, "Hello, Twippers. I'm studying the 3rd and final year of the biomedical scientist program at the Gothenburg University, Sweden. I've just spent my work studies at a medical parasitology lab where I introduced the old-timers to TWiP. Naturally, they became hooked immediately. Listening to you guys really made it easy and fun to learn the theoretical part of working in a parasitology lab, so I just wanted to say thank you. P.S. The temperature here is a chilly 3 degrees centigrade with rain."
Burr.
Wow.
Right.
It's cold everywhere in the world.
Right. That's 3 degrees centigrade, by the way. So that's
it's not
above.
freezing.
No, it's just above freezing.
Somewhere.
So that's why it's raining.
Somewhere I read
snowing.
an email where it was 3 degrees Fahrenheit. That is very cold.
That's cool.
That is painfully cold.
You bet.
Do you want to call it a day? Are you tired, Dickson?
No, I'm not tired, but I think
Listen, can we try and do this more frequently?
we
Like
can try
once every other
We
week?
can give it a whirl.
Because I have a lot of emails because people...
This is good. They flooded
Responded. It's
the
great.
e-waves with email.
All right, we'll do it next time. That's TWiP number 68. You can find this, as usual, on iTunes and at microworld.org/TWiP. And if you like what we do, I would like you to go to iTunes and rate the show. Leave it some stars and a comment. That helps to keep us visible there so more people will find us. We need more subscribers, Dickson.
We certainly do.
And we do love getting your questions and comments. Please send them to TWiP
And your corrections.
at twiv.tv. And I know Dickson gets most of them because he does all the talking. Dickson Despommier can be found at trichinella.org, medicalecology.com. Is it a .com?
Yes.
And verticalfarm.com.
Actually, it's medicalecology.org.
And verticalfarm.com.
Yes.
Thank you, Dickson.
You're welcome, Vincent.
And our special guest, Sagi
Oh,
Shapira,
thanks, Shagi. It was great having you on.
is at
That was
the
fun.
Department of Systems Biology
Listen to that.
here at Columbia University.
That's
Thank you, Sagi.
Really fun.
You want to come back when the priest is on?
I do.
Excellent.
I'll
fun.
come back whenever you want me.
Superb. Oh, great.
That'd be great.
No,
We like having chat.
We do.
We have a good
And I'll make
conversation.
sure Dickson keeps his distance.
I know you did that. You were good about that today. Very good. Thank you. I'm Vincent Racaniello. You can find me at virology.ws. The music you hear at the beginning and the end of TWiP is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. And I did ask him for permission to use his music on our show. He was
bet.
very good about saying yes.
Oh, yeah.
You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.